This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 737, recorded on March 30th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Good afternoon, Vincent, and all ships at sea. It's a beautiful day out there, although it looks nicer than it really is. It's a little windy and it's a little cool, but nice spring day. Cloudless sky and 14 Celsius here. Very nice. Yep, brisk. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 65 degrees, partly sunny, and that's 65 Fahrenheit, which is 18 Celsius.
And from Austin, Texas, Rich Condit.
Howdy. It's 76, headed for 82. It's cloudy. It's going to be cloudy all day. I can handle it.
And from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. We have 63 Fahrenheit, 17 Celsius.
All right. We have a few guests today who are all in the New York area. Let us start from Queensborough Community College, Monica Trujillo. Welcome to TWiV. Mispronouncing TWiV. Hi, Monica.
Hi, how are you?
Did I say your name right? Trujillo?
Close.
What's the right way? Tell us.
Trujillo.
Trujillo.
I am from South America. I am originally from Uruguay, and we pronounce the double L a little bit different from all the other Spanish-speaking people. It's a contamination with Italian. So
it's a It's a nice change.
Can't be Italian can't be a contaminant.
You've been enriched with the Italian.
My wife and I have traveled in Argentina, and they do the same thing there. And it was off-putting to begin with. And I said, "Oh no, that sounds lovely." Trujillo, right?
Yeah.
So we recently had someone on from Ecuador, from Forest Rowers Lab.
Yes.
Right?
They don't do that in Ecuador, though. I don't think.
Marisa.
What's her name? Does anyone remember?
Yes, Marisa.
Marisa what?
Marisa Rojas.
Rojas. Okay, Marisa Rojas.
So
Queensborough Community College, and you. I did a talk there recently, and that's how we connected, right?
Yes. So I am a microbiologist by training, but a bacteriologist by training. But the pandemic and my close friendship with John Dennehy at Queen's College and Davida Smith at the New School brought up this team that has been working since the beginning in Wastewater.
Okay. So also joining us from Queen's College, John Dennehy. Welcome to TWiV.
My pleasure to be here.
So we have Queensborough Community, and then we have Queen's College. And then our third guest is from the New School, Davida Smith. Hello.
How's it going? I'm a microbiologist from Dublin, Ireland, and I'm at the New School, but I'm actually a former colleague of John and Monica's at CUNY. I was at CityTech for a while, but now I'm at the New School over at Union Square.
Cool. So we have met a couple of times, Davida, right?
I think the
last was at a conference at Mercy College. Is that right?
That's right. We held up some giant microbes in the lab. It was fun.
That's right. I have pictures with you. Yeah. So the New School, but weren't you somewhere up there as well?
Yeah, I've been around.
Okay.
I was at CityTech. So I was at CityTech, then I was at Mercy, where I was the chair, and now I'm at the New School, which is right by Union Square, if you all know it. It's less famous for science, more famous for fashion. Parsons.
Okay.
Vincent, we very much appreciate your effort for reaching out to undergraduates. You might not remember this, but last year, about mid-April, we had invited you to give the keynote speech at our undergraduate research day. And unfortunately, we had to cancel that because of the shutdown that we experienced. But your graciousness was very much appreciated.
This was at Queens College. I agreed to do this.
That was true, yeah. You don't remember, I guess.
No, I don't. But if you want me back when it's over, I'll be happy to come.
Sure.
Yeah, Queens College is where in Queens? Exactly?
In Flushing.
Flushing, the former home of the Mets, or maybe they're still there, right?
No, of course
they're not too far from the Shea State, well, what used to be Shea State.
Yeah, City Field now, right?
City Field now, yeah.
So Queens College is famous for one thing that probably a lot of people don't know. Barry Commoner used to be there, a very famous ecologist, but it's the home of Louis Armstrong's music archive.
Mm-hmm.
Everything he ever did, everywhere he went, all his videos, all his music,
at Queens
it's housed
College?
in Queens College.
Wow. That's
cool.
Because that's where he lived.
And
Now,
we thought it was famous for John, you know?
And for John.
For me, this is very exciting to have some local schools here on TWiV. We don't often have that. And some in the outer boroughs, although New School is in Manhattan, as you say, right, Davida? Yeah.
Right.
So, Monica, where is Queensborough College in Queens? Is it also in Flushing?
No, it's in Bayside.
Okay.
It's about 10 minutes drive from Queens College, unless you hit traffic.
Yeah, which you always do, right? Okay.
And New School is in Manhattan, right?
Mm-hmm.
Yeah, it's right by Union Square. I'm around the corner from Union Square. So
yeah, Union Square is a cool place. There's some diner there, famous diner, right? Union Square Diner.
Yeah, that's gone a while. Yeah.
Really?
Yeah. It's pretty bleak at the moment. A lot of businesses have shuttered in the last few months. Yeah.
Okay. So now, Monica, you told us you're trained as a microbiologist, right?
Mm-hmm.
Where did you train?
Well, I trained in Uruguay for undergrad, and then I did my PhD in a program in combination with University of California in San Francisco. And so I did my thesis work over there. I worked some time in the biotech industry with streptomycetes, and then I kind of moved to New York because of family reasons, and then I ended up working at Queensborough, where, you know, in a community college, your chances of doing research are relatively limited. But working with streptomyces was good for me because I had, you know, they don't have very complicated requirements to grow them. And so I did some work there. But then when the pandemic hit, I was teaching a general microbiology course that had undergraduate research in it. And I had to change and decide what to do with this group of students that we were meeting every day. And so we switched to follow the SARS-CoV-2 story. That's what we did. We read every single paper that came out. And that's when I read this paper from a group in Denmark that was looking at the presence of SARS-CoV-2 in wastewater. And knowing John, I reached out to him and said, do you think we can do something like this? And he said, oh, I read the same paper. I was thinking about it, but how do we get in touch with the DEP people, the wastewater people, who usually are not very keen on sharing their
waste?
Their waste. Yeah. But luckily, through a combination of contacts that we had and everything, we were able to get that contact, and we started getting the samples and developing a protocol to detect the presence.
Okay, John, can you tell us a bit about your training?
Well, I grew up on Mutual of Omaha's Wild Kingdom videos with Merlin Olsen. So when I was starting out, I thought I wanted to get into behavioral ecology, and I actually did a master's degree on pronghorns, and that was at the University of Idaho. That experience, I enjoyed it very much, but it convinced me that getting funding for behavioral ecology is exceptionally difficult. So I started to think about alternative study organisms, and I settled upon mosquitoes because they're such a huge public health concern. And I did a PhD at Clark University. While I was there, my advisor on my committee suggested, where you're going in the future, you should model your career on Richard Lenski. And at the time, he was very famous for his, I think it was 10,000 generation E. coli evolution experiment.
Yeah.
So I reached out to Rich, and he said, well, I don't have any positions at the time, but, my postdoc, my former postdoc, it might be a PhD student, I'm not sure, Paul Turner at Yale University is looking for someone. So I reached out to Paul. Turns out we were able to get funding for a position for me at Yale University. And I discovered bacteriophages, and the rest is history. I just, fell in love with phages. And ever since then, I worked on phages. And maybe about 5 or 6 years ago, I started thinking about working more with mammalian viruses. So I started out with influenza and rotavirus and trying to incorporate them into my research. And I've long known Monica for quite some time. And now I realize that, I've been pronouncing her name wrong all this time. But, she, like she said, she reached out to me last spring. And I immediately said, that's a great idea. I had been, searching for a way that as a virologist I could contribute to the pandemic that was striking New York City at the time, right around the end of March, early April. This is when we started our discussions about how we can help our city out in this time of crisis.
Now, why did Monica reach out to you? Just because you knew each other, or do you have lab capabilities that could be used?
Well, we had collaborated on a soil microbiome project. I had looked at the soil microbiome across an urbanization gradient spanning, New York City out to the tip of Long Island. And basically we looked at bacterial diversity and fungal diversity along the span. And I had invited Monica to join me in that work. This was probably about 5 or 6 years ago, right, Monica?
Yeah. But also it's true that John's lab has the capabilities to work. He has a BSL-2 laboratory, and he, he had all the connections to convince the people at Queens College to allow this work to happen. Yeah, absolutely.
Got it.
John, when were you at Yale? I
was at Yale from 2003 to 2006.
Way after you, Rich.
Yeah, I'm kidding. That's a common theme now.
Yes.
Way after me.
It's not going to get any better either.
I know.
So John, I'm really Paul Turner was on TWiV with Nels Elde maybe 2 years ago. Terrific phage therapy, right? Among other things. And the other thing,
I knew him before he got tenured. I used to tell him, if it wasn't for me working in a lab and publishing all those papers, you probably would have never got tenured.
That's great. I also want to point out, John, I'm very happy to see not only Fields Virology behind you, but what I think is Principles of Virology, a few places over, two volumes,
right?
That's
right, yeah.
Thank you so much. I'm glad you don't just have Fields there. That's great.
Ask him if the bindings are broken.
Hey, Davida, how did you get involved? Well, first of all, tell us your training. You mentioned it a bit, but elaborate, please.
Well, I'm the outsider, so I'm the foreigner. So I'm from Dublin, Ireland, and Monica as well. The two of us we're the immigrants. So I'm from Dublin, Ireland. I went to Trinity in Dublin. I did my undergrad and my PhD in Trinity in Dublin. And I'm a microbiologist, so I studied staphylococcus actually from animals. That was my undergrad. Molecular epidemiology of staph from cows with mastitis and chickens and goats and things like this. And when I came to the States, I worked on MRSA evolution. So I was at NYU and I was at NYMC and I was in UMMC for a bit in Mississippi. And then I've been an assistant professor, associate professor, and now associate professor at the New School. So my research was always in staph and microbial genomics. But when I started working with undergrads, I had to transition from scary MRSA to non-scary staff-related projects that were undergrad friendly, frankly, and affordable, frankly. accessible. So I've started working in the built environment. I do a lot of microbiome work in the built environment, studying staff dynamics in the built environment. Very simple stuff. But I'm a huge fan of John. So I've known Monica for a long time because we've gone to various conferences together from an education standpoint. But I used to follow John's career, and I met John at a workshop. I was sitting beside him, talking about this fabulous virologist that I had to meet. That I was really looking for. Do you remember this? Really looking forward to meeting this guy, and I was looking out for him, and he just turns his name tag over. He let me talk about him for about 20 minutes before he says I'm John. So I've known John a long time as well, and when John heard from Monica, John got in touch with me to bring me into the project. So I bring sequencing in. So I have a sequencer that I've been using to do microbiome work with the students here. And it's an iSeq, so it's an affordable sequencer, benchtop sequencer. So we thought the three of us together could do a good job of investigating the wastewater dynamics through sequencing and real-time PCR.
Okay.
Davida, I read through your little bio online, and I just have to point out one of your freshman seminars that's amusing to me. It's called How the Toilet Changed the World.
Yes.
So I thought, oh, okay. So now I know why she's in this.
Well, I love poop. I do love poop. It has to be said. So I think that's partially why they brought me in. So my father is a plumber. And so it was my father that actually got me my first job. And so everything that I do stems from all the experiences that my dad shared with me. So I love, at the new school, we have a very strong social justice mission. And so myself and Monica, we totally see eye to eye with respect to social justice. So the new school's curriculum is all tailored to kind of a social justice framework, and how the toilet changed the world is all about cholera and sanitation and prevention of disease. So, yeah.
John Snow.
John Snow, exactly. He's my hero, of course.
Oh, yeah, mine too. I once had a drink of water at his pub in London.
Oh, wow.
I walked in, I didn't want a beer. I said, I want a glass of water. The guy says, you want water? I said, yeah.
Put the handle back in the pump.
Yeah, exactly right. Exactly. By the way, I think you should move to Flushing.
Yes, I should. Yeah, Meadows
Dixon, you always said that the toilet and sewage was the greatest invention of humanity. Right?
The outhouse in the rural areas and the flush toilet changed the world. It certainly did. Yeah. For better and worse.
For better and worse, but I bring it one step further, and I train my students how to make money from poop. Biogas.
Hey, I'm a parasitologist. I'm on your side.
Davida, is the new school a primarily undergrad institution, or do you have graduate students, or kind of how are things set up there in the sciences?
Yeah, it's a little bit strange. So I'll just tell you, I'm one of, there's only 3 scientists actually in the department. There's 2 biologists and a chemist. We're really small. So Eugene Lang is the liberal arts college of the new school. There are kind of 6 institutions that make up the new school. And the sciences program is tiny. We have an interdisciplinary science program. So it's not majors. The students get a very broad kind of training. And we're kind of thinking that students might go on to do policy and kind of, social fronted work that might propel science, if you like. So it's a little bit different. But I was recruited to do undergrad research with students. So we don't have science graduate programs. We have psych and things like this that do have graduate programs, but we don't in the sciences. Cool. It's an unusual school. It's very different.
I have a good friend who teaches there. Her name is Michelle Laporte, and she's an environmental urbanist.
Oh, yeah.
So I think I would encourage her. She's a delight to talk with, and she's broadly based. And I give an annual lecture there on something that I'm not. I hadn't been trained for it. I picked it up after I retired, as it were. but I've enjoyed going over there. It's a very busy place that's got lots of activity, and it's got a rich history. You guys, that's a great school. A great school.
So Monica, let's go back now. You said last year about the onset of the outbreak in New York City, you had this idea to look at sewage. So tell us how that started.
Well, it's funny. I was just thinking about that. In particular, Queens and minorities were severely hit in New York. So as you realize, I'm a Latina, and I live in Westchester, that it's kind of an affluent suburb, but it has some small towns there have a high proportion of Latinos. My students, the ones I was teaching, were severely impacted by the disease. And I found myself, and John can contribute to this too, I found myself being the person that they could talk about the disease and what to do and how to protect themselves, because I had time in my hands, because I was only teaching online at the beginning. I contributed to food distribution, to training people to how to wear the mask and how to keep, the social distance, what it meant. And I really found that my scientific background gave me the tools to try to find another way that was not dependent on the clinical assays to detect the disease and to try to figure out what was happening and why certain groups seemed to be more fragile compared to others. So for me, I like to say that it was an experience that I never thought I was going to have on my life. To be really honest, I had a scientific career that, because of not being in a research institution and mostly dedicated to education, had never had like any real potential to impact directly somebody's life or to do something that seemed like on time. So it was, I really have to acknowledge the pressure of my student and society on me to try to use my background to do something that, as John said, could contribute. I share with Davida and with John a very well-defined sense of justice and equity. And the pandemic was really not fair. I happen to have a son who's a doctor and who was at Columbia Hospital helping people to make the last decisions, because he's a psychiatrist and he was redeployed and he was in very close contact with the Latino community. So all these things gave me this interest to learn about viruses at a speed that, was not what I was used to do, right? And to find partners in research like John and Davida made a huge difference. I'm somebody who really likes to collaborate, and this was a unique opportunity.
Yeah, I remember that sometime last year, a neighborhood in Queens, Corona was one of the hardest hit. They had the highest seropositivity,
Exactly.
right?
Yes.
One of our technicians is from that neighborhood, and he was unable to go shopping. And I offered to bring him some groceries because his whole household was infected and they weren't able to leave the house. He said he wouldn't even meet me in his neighborhood because he was so afraid that I would get infected just by coming to his area of Corona. We ended up meeting at Queens College where I gave him some groceries, but at that time, Corona, ironically, was the center of the epidemic in the United States.
Yeah, it's amazing how I couldn't write that into a movie, right?
Yeah, exactly.
And they'd cross it out.
So Monica, you got the idea to look in wastewater. So how did you? And the DEP doesn't want to give you wastewater. So what did you do?
I have to give them, credit, because I got in touch with somebody who gave me a name from DEP, Pamela Ardo, and I sent her an email at 6 p.m. One afternoon. And by 8 p.m., I had a response from her. And she said, we are interested. Tomorrow morning, you are going to get an email from somebody that is going to contact you. And so John and me and Davida, we met with this guy who's an engineer. And he had, I don't know, you guys can correct me, but, superficial understanding of what we were talking about. And he wanted us to be his backup because they had already established a collaboration with some more prestigious universities to develop a protocol to detect the genetic signal of SARS-CoV-2 in water. But he said, you guys are CUNY. We are all from the city. We'll give you water.
If you waste
Monica,
wastewater.
you left out the part where we emailed about 2 dozen people asking if we could access wastewater. And we, for most of them, we did not even get a response back.
Yes.
That's incredible.
I'm not surprised, given the bureaucracy of New York City, that you even got it done at all, right?
Yeah, it was just, I think that it was just that we could get this woman who's very pragmatic, right? And they had a soft spot for CUNY because we were really the backup. Right?
We're the little guys.
Yes.
Yeah, but now you're on TWiV and they're not.
That's true. Exactly. So
the story has been better.
Department of Sanitation has had
Dixon,
an issue.
Wait, hang on. She wants to say something.
I'm sorry.
Go ahead.
The story is even better because when was it? July or something like that? We get an email saying, can you share with us your progress? And we're like, sure. And then I said, okay. So we present. I said, can you share with us? And they said, no, we can't. So there was a silence. And, I do have some Latino genes. And I said, okay, forget everything we have said. But if you cannot share with us, we cannot share with you, or something like that.
Nice.
But the next day, there was another meeting. And we found out that their protocol had not worked.
Mm-hmm.
So they wanted us to tell them. So
good. Dixon, what were you going to say? Sorry.
I was just going to say that there's a history of a bad press with regards to the sanitation department because every spring and fall, when the lakes up in New York State turn over, there's a disease called cryptosporidiosis. And that organism has a way of getting into the drinking water. And at that point, they got so much bad press about New York drinking water that they just wanted to not do any work with anybody on the outside. It's bad press. New York City has very good drinking water, but the perception was in some cases that if you drink the water, you're going to die. So, the press sort of gets the stories out of control. And that's where they're coming from on that one. And I'm sorry that you got embroiled in it, but they've been very close to the vest in terms of keeping all that to themselves. You're absolutely right.
So I noticed from your paper here, if I understand it correctly, that you have, or you at least report in this paper, samples from 14 different treatment plants. Mm-hmm. And you say
that's the number
they're in New York City. Okay, that's a big place. Are they like all over the place?
The distribution of the plants, Monica.
Yeah, it's all over the city. Each borough has, depending on the size, has 1 or 2.
Okay.
Yeah, there are 14.
Monica, tell Rich how much water New York City throws away every day.
I don't know.
There's about a billion gallons passing through.
It's 1.2 billion gallons.
Yeah.
And they wouldn't give you 1 cc?
Well, this is the drinking water folks and the wastewater folks are different.
No, I appreciate that, but still.
I'm glad they're different.
But they're sharing it all with us now, so they've been very clear with that.
That's good. That's good.
Give them,
So at some point you got samples. So how did this work? Did you go to pick them up? And were they like little tubes or huge? I'm curious as to the logistics.
I can bring one out and show it to you if you want.
Yeah, sure.
I have one in the lab. Give me 1 second. Yeah. Monica, talk and I'll go get one.
So it started with me going to Newtown Creek to pick up 500 ml bottles, meeting John at the door of Queens College, right? Because I am not a Queens College employee. Now I have no problem. John worked everything out. But at the beginning, I couldn't get in. So John will bring his car to the entrance of Queens College. I will be there. It was on Fridays, right? And we will transfer coolers right? During this time, John was developing, yes, that's exactly.
Lovely.
So during that time, John developed the safety protocol. It was approved by Queens College. And then I was able to get into the building and we started working. Until now, I go every Monday. Now we changed to Mondays because Friday traffic is worse. And we pick up the wastewater samples I pick it up every Monday.
Okay. And you pick it up from
Newtown Creek. Newtown Creek.
So what is Newtown Creek? Tell us.
It's a microbiology lab that the DEP has. All the samples are delivered there for them.
Okay.
And they aliquot. And then they do the analysis and we do the analysis. And we provide them with our data that, and they do their work on their side. And they look at the two sets of data and then they inform DOH.
Okay.
If you're ever driving down the LIE as you're heading toward the Midtown Tunnel,
It's got the
look on your left-hand side, there's a giant silver dome.
That's right, the digester.
Newtown Creek, right on the Gowanus, right?
It's the newest of the treatment plants, I think. But what stage of the sewage do you get?
We get the inflow.
Yeah.
So
The inflow.
unprocessed.
Unfiltered input. Oh, wow.
Yep.
So then how do you actually deal with that?
Yeah, that's my next question.
You actually doing your experiments and how
a
do you
BCL4 facility.
Yeah. There's a lot of safety and extraction that I can imagine has to happen here.
So that's where my phage background came in handy because I've been used to, using wastewater to isolate bacteriophages against bacterial pathogens, the escape pathogens. So, we were familiar with trying to, concentrate phages. So we tried the same techniques on SARS coronavirus, assuming hopefully that they would work. And as it turns out, very similar.
But you start off with the pasteurization, the safety procedures.
Walk them through the procedure, Monica.
Yes. So we start by doing an hour exposure of the bottles that are closed at 60 degrees, right? And after that pasteurization, what we do is we centrifuge and then we filter and then we add the PEG. We have, very good laboratory techniques. We had been, during all this time, we have also been doing, it's not clinical testing, but we do take saliva from everybody who's working to make sure that, and we do the test, and we have frequently got tested. Nobody, since we started working, ever was positive. So I think that the fact that John had people who he had trained, right, that he was able to bring over to work with us made a huge difference. Mm-hmm.
So after the pasteurization, you mentioned centrifugation and then filtration. So that's to get out different size particulate matter, right? And then you're using the filtrate to precipitate. I just have to interject something personal here. Back from my phage days, working with Ed at Yale, okay? When he talked about centrifuging something to get out debris, he would talk about spinning out the poopies, okay? And in this case, it's quite literally spinning out the poopies. And then you do, and I was also quite amused, you do this PEG precipitation, which is also from my phage days. We used to grow up phage in 50-liter carboys. And with a volume that big, the only way you're going to concentrate it is with a polyethylene glycol precipitation. Okay. So the thing that has puzzled me all through this is I have trouble imagining you've inactivated the virus. I'm trying to figure out what sort of particles are surviving all of this, because I don't imagine that they're intact SARS-CoV-2 particles. It must be something like nucleoprotein or something. Do you know?
It's possible. We don't really know for sure. But one thing we do know is, I was required to pasteurize before we could even open the bottle.
Right.
There's a condition for doing this lab, work in my lab, which is BSL-2. Technically, if you were to research on SARS-CoV-2, if you're working with live virus or amplifying the virus, you need a BSL-3 lab. We were able to convince our safety people that, we would not even open the bottle until we pasteurized. This had the added effect, actually, of increasing the SARS coronavirus genetic signal. We were able to get better recovery from pasteurized samples than we would from unpasteurized samples.
I have to believe that it must be a nucleoprotein of some sort, the nucleocapsid. That makes the most sense to me. These procedures must destroy the membrane, I would think.
Yeah.
There's something odd going on, because we cannot replicate that same result with bovine coronavirus, which is extremely similar.
Okay.
The pasteurization does not assist with the recovery of bovine coronavirus. So
I love that
we have experiment designed to try to better understand this, but at the time where, right now we're, really deep into sequencing.
Hey, it works. You don't ask questions.
So Monica, you've mentioned we and us a few times. I'm just trying to imagine this kind of, how big is the team that is working on all of this? Is this technicians? Is this undergraduates? Who's working on this?
So it's a very unique team. I think the only one who was born in the United States is John. So we had the undergrad. So these are students who have graduated from Queen's College, but they had worked with John. So John brought them back. So one of them is Zach. He is originally from Nepal. And then
Myanmar.
Sorry?
Myanmar.
Oh, okay. Sorry.
Burma.
Okay, Burma. Yeah. You're right. Sorry. And then there are two other bachelors who are originally from China. And then there's somebody else who's also has contributed a lot, who's a postdoc, who is from India. So it's been your typical Queen's immigrant story.
And they're all working in, the space limitations are still there, and everybody's working kind of in shifts. So that's really important to understand as well. So everybody's working over there in shifts.
Where does this happen? Do the samples go to John's lab first?
Yes. Everything happens at John's lab.
Okay. And
then I drive our samples across the city to the new school.
Okay. For sequencing.
Yes.
And then you process them for sequencing, Davida?
Yeah. So in essence, we're still kind of, we're still working at the kinks of the whole genome sequencing, but we're doing targeted sequencing. That's what we have in the paper. Mm-hmm. So in essence, when Monica processes the samples, she'll either bring me RNA or cDNA, and then I do the sequencing. And so we're trying a bunch of different protocols. We actually got some funding through John from a QIAGEN grant to help us do some sequencing with QIAGEN's materials. We're using Swift's protocols. And then we're doing our own targeted sequencing approach, which is the one that's working the best for us so far. Okay. But we're doing it on the iSeq, on that little sequencer.
Got it. So, by the way, I haven't heard the word pasteurization used in years. It's very curious that you're using that, because you're using the same conditions, I would guess, as Pasteur would use to clean up milk, right?
What we're absolutely dying to do is put some of those samples into cells in the BSL-3 lab, but we haven't got access to one.
Well, there is one here, and could be arranged if you guys want to talk, because I have a person who is trained to work, who you met at Mercy, Amy Rosenfeld, right?
Yeah.
And so let's chat offline, because I was going to ask you, I'd love to know I cannot imagine, as Rich said, that there's a lot of infectivity in there, given what it's going through. But nobody has really looked at it. Yeah,
nobody has because of the safety protocols. But also, John has access to the dorm samples, which could potentially be fresher. And if we can get more fresh samples, we could do some analysis on that.
Okay.
Yeah.
So there are 14 treatment plants. You're only getting it from the Newtown facility, or
no.
all of them?
We are getting from
All
all.
of them.
So every week you do 14 protocols for this of 500 mils each.
Yeah, we are now with 250. But
250.
And originally, Monica, you were getting them twice a week.
yes, we were getting them twice a week. We also got some from Westchester at one point. But the truth of the matter is that we didn't have the women power to process all of that.
I can imagine.
It was just not possible.
So is your data going to shake out areas of low infectivity versus high infectivity because of the distribution of the power of the treatment plants?
We do see a difference, clearly.
Yeah.
Clearly. And also, we are also interested in detecting, even now, influenza, and we also see a distribution pattern, right?
Yeah.
So certain areas of the city don't have it, or we cannot detect it.
Is there an ethnicity or a economics to all this?
So that's where we cannot really talk. We can't speak to that yet. For reasons of privacy and such, we have to protect the identity of the plants. But we know where the cases are, and we can overlay the counts.
Right, exactly. Right.
So we want to bridge those two things together, but we have to effectively persuade the Department of Health that this kind of work is warranted and needs to be invested in, so that we can continue it. So we need to get more money.
So you have been able to quantify, have you not? So what sort of quantities do you see? What sort of range?
Influenza or coronavirus?
SARS. Coronavirus.
What are the quantities we've seen? About 22 or 23? So it's very, very high quantities, I think, in January.
So in terms of genomes per liter or copies per liter, how does that translate?
Let me look at the data because I don't.
So while you're doing that, John, do you do any readouts in your lab? Do you do PCR or something to quantify?
They're doing RT-PCR.
That's right. So we have we do a standard curve using a synthetic SARS-RNA genome, which is provided by a company named TWiV Bioscience.
Okay.
Now IDT.
Got it. Okay. What are the primers you're using for?
They target the N.
N protein only?
Yeah.
N protein.
So I'm looking at the paper we published, and we said it was roughly about 1.5 million copies of N1 per liter.
Yes.
And you multiply that by what did you say? 1.2 billion gallons per day. That's how many viruses are flowing through New York City.
You might say it's a shitload of virus, and you would be right.
So do you have some sort of a negative control, like a pre-pandemic sample, or what do you use?
No.
That's a big regret is not having some pre-pandemic samples to compare to.
They don't save it? They don't save any samples?
We use no template control. We have a positive control.
We have a method blank control that we process in parallel to make sure that, this virus, and I'm really fascinated by this. We literally don't have the time to try to answer these questions. But as John said, the bovine coronavirus it seems to be way more fragile than the SARS-CoV-2, right? You know how RNA, it's technically, you have to keep it at -80 because it degrades so fast. Not at all, right? We keep it at 4 degrees, no problem, right? The only thing that we do have seen is that if you keep the wastewater at -80 and you then defrost, you do lose a significant amount of signal. Yes, that is a fact. But we don't know what can explain this robust behavior. We imagine that because SARS-CoV-2 is shed in feces, that perhaps, and then they stay in wastewater, they perhaps attach themselves to solid as opposed to a spike from the bovine. But we don't really have time, or we haven't had time to develop an experiment to answer this.
But you're working on it, Monica.
Yeah. But
so has every sample you've checked since the beginning been positive?
Yes.
To some extent, yes. You're never going to have a negative, as far as I can tell, for as long as this virus ain't going anywhere. So it's just too bad you don't have any pre-pandemic samples.
I wonder if you could get samples from some other area in the U.S. or elsewhere as a negative.
We did because we had some samples from the northern part of Westchester when we started, where there was no cases, and we definitely did not detect it.
So each of these treatment plants represent how many people?
Could be anywhere from around 100,000 to several 100,000.
Okay.
And they're also, remember, in New York City, we have a unique setup because we have a combined sewage overflow system. So we're unique in terms of wastewater infrastructure across the United States as well.
You really are.
What does that mean? Tell me what that means. I don't understand.
So effectively, the storm drains also flow into the sewage lines across the city. So snow, rain, all of that will impact.
So then you get SARS-CoV-2 virus in the Hudson River directly.
Yes, you do. We've started to sample Gowanus Canal because John and Monica and myself, we also looked at Newtown Creek. So we've just started pulling some samples from Gowanus, and we intend to look at those as well.
Right. So New York City ships all of its wastewater to one place in somewhere near the entrance to the Long Island Sound, and they centrifuge all the sludge out. Then they redistribute that back to the same boats that bought it, and they bring it back to and then they treat it with chlorine and throw it away. Do you know how many billions of dollars we spend throwing good water? If we had the water and we could purify it and drink it, it's the most valuable liquid on the planet. And here we are, we call it wastewater, but I think that's a wrong term for that one. Just unrecycled fresh water.
So can you give us an overview of what you found, first in terms of prevalence? I mean, peaks and troughs over the last year. Can you do that?
Yeah, John?
Sorry, what was the question?
So if you just look at every week, do you see levels going up and down? Does it correlate with cases, for example?
Yeah, it very closely matches the clinical cases. What we saw when we first started for real, probably last June, we saw a high level of cases, and it continued to kind of decline across the summer, and reflected what we observed in New York. And then probably about mid to late September, we observed it started increasing again. And it continued to increase until the end of January. And I think that's when it started declining again. But it very, very closely matched what we saw in the clinical cases in New York City.
And right now, is it kind of plateauing?
It is kind of plateauing a little bit. Monica thinks it's going up again.
It's right now.
Unfortunately, it varies a little bit, but it's creeping up a little.
Okay. Yeah. Okay. Yeah. And you're going to keep doing this forever? Is that the plan?
We've been told we'll have funding at least until the end of April. And we hope the DEP will choose to continue doing this research because I do think we provide something valuable to the city of New York.
I think if you should do it forever, because even if there are no clinical cases reported, you may still see a low level of virus, which means it's somewhere in someone asymptomatically. I think it's incredibly valuable to do that. And
we agree.
Exactly.
And we wanted to do it, like our original plan was to do more than just the surveillance. We wanted to look at the epidemiology and the evolution of the virus in the asymptomatic population, right? But in essence, for that, we just need to get funding beyond April.
Is there no chance of getting EPA funding for this? Because this is really at the nexus of all of the other problems. You can be a canary in the cage for the outbreak before anybody else knows it.
Not only that, we have through CUNY, we have the massive infrastructure of CUNY to put behind us.
Yeah, that's exactly right.
We have a group of schools that are full of students who want to do something for the city. And if we had the mechanism to train them, they could be our future technologists who are doing surveillance for the city's future outbreak. So any funders who are listening and want to help with the city, consider donating to CUNY so that we can train the next generation of technology.
Yeah.
Now, Davida, you said you do some targeted sequencing. What are you targeting? The spike, I presume?
Yes. So Monica and Sharon, the postdoc that Monica was speaking about, they designed a primer set that have Illumina adapters on them. So in essence, it's a short enough fragment that we can sequence it directly on the sequencer. It's using the same technology you'd use for the microbiome. And then we basically we just analyze the FASTQs that come off of the sequencer. And we look for variants in the FASTQs I'm using Genius software, which is a commercially available software package. And then we look for clinically relevant variants that have been described in the literature. But we're also looking at the variants that haven't been described in the literature, and there are many of them. So our preprint that we shared with you all describes some of the other variants that we've seen. And that with more funding, we could we want to do this every week so we can do trend analysis. What are the variants that are ebbing and flowing in the population over time? So we could see if we're seeing some new variants that are New York specific. But also Monica has in the pipeline additional regions that we're going to sequence as well. And as she said, we're expanding now into flu. So we think that this it's a very effective way to monitor viral dynamics in a population of asymptomatic folks. Sure. So there's huge value in doing it.
We've been very focused on sequencing ever since last May. And this has not gone over well with funding agencies. We applied to NSF and NIH. And, the comment was, we don't think you can sequence SARS coronavirus isolated from wastewater.
So I have a question about that because you have been able to do the sequencing using this protocol, but as your paper says, you recommend removing the pasteurization step if that's the main goal. So
I actually think our latest data shows that the pasteurization doesn't have an effect on sequencing. But at the time we wrote the paper,
you thought that it did.
We wondered whether it could possibly negatively impact the sequencing.
Okay, because I was wondering if there's any way short of working in a BSL-3 then that you could work with wastewater.
Right.
Probably not. Okay.
Yeah, we don't see a difference there, I don't believe.
Okay.
But one of the major concerns with the whole genomes, and remember, there are tons of labs around the world that are doing this as well with varying degrees of success. Some labs have been able to do it wonderfully. The issues are always going to be coverage of the genome. So there are these gaps across the genome that are just not sequenceable for whatever reason. When we did our initial pilots, I couldn't get whole genome coverage. But when we do targeted sequencing, we get lovely depth of coverage of the specific region that we're interested in. So from an economic standpoint, but also from an informational standpoint, we go with what we are getting the data from, and the targeted approach seems to be working for us right now. So there are some labs who are doing kind of multi-locus approaches where they're looking at multiple regions, getting lovely depth of coverage again. And I think that's probably going to be the most useful approach until we get the whole genome up and running really, really well. So we're trying to combine the two, if you like.
Yeah. You ought to look at some other sites besides Spike. There are a lot of interesting loci. I think Orfate is really interesting, and in some areas it gets deleted, and that would be really cool to see what's the situation here in New York City. I don't think anyone's looked at that. Well,
if you want to collaborate, Vincent,
Yeah, let's talk offline. We'll talk about that for sure. And eventually you want to do whole genome. Is that the plan?
Yeah, I mean, we're doing it right now. We're trying to. So again, we have the SWIFT protocol. We've been able to sequence the genome. We just didn't get very good coverage of the genome.
Now, is anyone else doing this in New York City? You said someone started in the beginning and then they couldn't get it to work. So what's the story? What's the story there?
There's the other group. It's part of, we do work with them, right? DEP has these 2 academic labs that support them, and we contribute. We have meetings together, and hopefully we are writing a paper all together, trying to identify the variables. What sources of variable are in this detection method beyond the difference in the concentration of SARS-CoV-2. And it's a really very interesting experience of collaboration, where you need to work with people who have a different goal, who have different means, who have different access to resources. But what guides all of us, and I think this is really fair to say, it's a real interest in serving the city.
Yeah. I have to confess, though, that most people were not interested in sequencing until December, when we started finding out about the UK variant, right?
Yeah.
When that came out, all of a sudden, everyone's interested in sequencing SARS coronavirus. So.
And then you see articles about variants in the New York Times, which we never thought we would see.
Yeah.
Right?
Don't let's not talk about that. In retrospect, we should have been sequencing extensively from the beginning, because this was an incredible opportunity in a huge outbreak to get evolutionary information. And why we weren't, it was triggered by the UK variant is crazy, but that's done. So
yeah.
Do you have a predominant variant right now in the sewage of New York City?
E484K is the most prevalent variant.
That's a single amino acid change in spike. Yeah. Yep.
Yep.
Yeah.
Okay. And that affects a monoclonal epitope, right?
Yeah.
Yeah.
So we're watching what we cannot do. The limitation of the method we're doing right now is we cannot necessarily always link all of the variants, because it's a short read, but we can at least connect 1 or 2 of them.
Got it.
That's what we're watching for.
Okay. So it's all
you can see whether or not there's a particular allele
on the same.
You don't know what the genome actually looks like that came from.
No.
Yeah, and when you do whole genome, you have the same problem.
Yeah, because it's all fragmented.
Right.
Yeah.
So
unless you do some long read, like what's that?
Minion.
Yeah, Minion. Yeah. Minion. You used to call it Minion. I love to call it Minion.
I like Minion too.
That's something that people from a clinical background sometimes tend to dismiss the results from wastewater. And I do think that they serve different purposes. They are looking at the different problem. And then John sent to all of us a paper last week where they said that most of the sequencing in clinical samples was done with samples from the upper respiratory tract. Yeah. But they think that some of the mutations or whatever happen at the lower level, and you don't have them unless you are looking at the other end. So I think it's an example of the need of using complementary approaches.
Well, we have colleagues in Sinai that were beginning to look at samples from fecal specimens from the same patient over time. So the last time we spoke to Karim and colleagues with Sinai, they were looking at within patient variability, but variation all the time. But we only care about the wastewater right now.
All right. Well, this John, go ahead.
California variant, and also there's a unique mutation associated with New York variant of interest. It's not quite a variant of concern, but and then we also have some of these mutations are associated with multiple variants, like E484K found in Brazil and South Africa, I believe.
Yeah.
There was another one. The other one was N501Y, which was found in the UK variant, the P1 variant from Brazil, and the variant from South Africa. I think the South Africa one is probably the greatest concern.
Yeah. All right. It's a fascinating story. Anything else we should cover before you guys leave? Did we cover everything you'd like? Yeah.
We, again, like we said, it's a call out to help those who haven't got access to all the facilities and resources across New York. So,
okay, from Queensborough Community College, Monica Trujillo.
Perfect.
Thank you so much for joining us today. I should say that, so you heard me give a talk to, was it Queensborough? I don't even remember.
Yes, it was at Queensborough. And
Yes,
I tested you because I sent a question and I said, do you think that it's useful to look at wastewater? And you say, yes. I said, okay, so we can talk to him.
You said you should look at it and then throw it away.
No. I think it's a great idea. And someone had invited me based on another talk I'd given locally also.
You've talked at MacCube as well. You've talked at MacCube a couple of times.
And you were there. I saw you in the Zoom.
I always follow you wherever I can. So
Just stalking.
Very good.
I am stalking.
Very cool. Very cool. Well, I visited you not too long ago.
You did. And I'm a legit fan, so you know.
And I've seen you at ASM meetings, I think twice.
Yep.
And then there was a Columbia meeting where you talked about your built environment work with your, which I think is good. You can get the students to do this sequencing, wipe things, and there's so many cool stories. Very good. And so Davida Smith from the New School, thank you for joining us today.
Thank you. Thanks for having me.
I'll be in touch. Are you in a common area? That's why you have a mask on?
I'm in the lab right now, so I have to wear my mask, and the students.
Is there anybody there? They're right behind you?
A couple behind me. Will I bring them in?
I'd like you to just take your mask off for 1 second. Sure. Hi. Thank you.
But always her Irish eyes are smiling.
They are.
And from Queen's College, John Dennehy. Thank you so much for joining us, John.
Thank you for having me. It's been a pleasure.
That was terrific. Absolutely terrific.
We'll follow this, and I hope you get funding to keep going. And I think it's wonderful. I really like, on many levels, like the community involvement, the collaboration, and the science. It's really cool. So
yeah, very good.
I'm glad we could showcase it.
Love it.
Super cool to hear about. Thanks, guys.
Yes, thank you.
Okay, I'm glad I got that to work.
Yeah, that's great.
Because that's a cool story, right?
Mm-hmm.
Yeah.
I mean, and it's local. Love it. Which is like, folks, if someone asks you to talk, just do it. You never know what's going to happen. There's no downside to it.
I definitely
not you. I'm not talking to you guys here. I'm talking about the greater audience. Yeah, I know you guys are all good.
I really want to reach out to them and sort of talk about working with students in the greater New York area.
Mm-hmm.
Well, you go to MacCub, right?
Sometimes, yes.
Yeah. What is it? Metropolitan? I don't even know.
I think it's Metropolitan Association of Colleges, college biology, maybe.
Undergraduate biology. Yeah.
Yeah.
Yeah. So, I've been to one physically in Jersey City. There's a university there whose name I can't remember. And then this past year it was online. Yeah. And it was was there one at Drew?
No, the one that you came to at Drew was Tri Beta.
I did a Tri Beta last week.
You did?
Jersey City.
St. Peter's, that's right. Yeah, I did a Tri Beta last week at Drew also, yeah. I like doing the local stuff. I think it's important, right?
Yeah, I do too. I enjoyed that when I was teaching. Very much.
Okay, cool. All right, a couple of announcements. We have a few things to chat about. I want to remind everyone of the Adenovirus 2021 Symposium. It's all online, of course, but you can have a good program. And I just registered this morning. I finally got past all the Dutch. I would go to pay and everything was in Dutch. No, I don't understand what this means. I finally figured it out and gave them my 95 euros. So I'm really looking forward to that. That's in June. So we'll put a link to that in the show notes. And PSA from the ASV vaccine town halls. Kathy, brief announcement.
I don't have much to say that people haven't heard before, except that we have these town halls where you can go sign up for a Zoom meeting, ask questions. They're 30 to 45 minutes with two experts in virology or medicine, immunology, various things. I don't know if Brianne, if you've done yours yet or you're signed up for one.
Mine is tomorrow.
Okay. So you may see people whose names you recognize. Either they've been guests on TWiV or they're co-hosts on TWiV. And spread the word.
And they'll keep going on for as long as necessary. If you look at the registration now, there are 14 of them currently, one from tonight through April 7th. And this roster of meetings, when they're given, they disappear. And when people sign up for new ones, they appear. So
cool. Very good.
Anybody can register.
And the website is asv.org/education. Or if you can only remember asv.org, it's on the homepage as well in 2 different places to get to the town halls.
I also want to mention the group we just chatted with. They have 2 MedArchive preprints. One is called Protocol for Safe, Affordable, and Reproducible Isolation and Quantitation of SARS-CoV-2 RNA from Wastewater. And then a second one, Detection of Mutations Associated with Variants of Concern by High-Throughput Sequencing of SARS-CoV-2 Isolated from New York City Wastewater. So we'll put links to those in the show notes. I thought we'd chat a bit about the WHO-convened global study of origins of SARS-CoV-2, the PDF of which was released today, and I got an embargoed copy yesterday under threat of execution if I shared it. But I do embargoes for lots of things, and I'm good with embargoes. So this is a long report which summarizes what you've heard. This is a group that went to China and spent a while, and they worked starting last year gathering information, and then they physically went there for a number of weeks. Had to do a 2-week quarantine when they got there, and they did Zoom meetings or whatever, and then they were able to go out and visit places, and then this is the result of that. And
this was back in what, February mostly, right?
Mm-hmm.
Yeah. So it is very readable. I suggest you download it and take a look. I think they've done a lot of work, and I don't agree with some of the criticisms of it, but I thought we could summarize it and what exactly they've done. For example, the idea is the information that's there, could they go through it and say, can we get information on where the outbreak began, how it began, and whether it began any earlier than December 2019? And so they looked at a variety of records. So this is 17 people from China, 17 other experts who are interesting, are not named on this document, although I understand they're elsewhere, right? Mm-hmm. Isn't that weird to issue a document and not have the authors on it? I think it's weird anyway. So
yeah, I think it's weird too.
I think they should have their names on it. For example, they had lots of samples that were stored, and they went through those and so forth. Anyway, so let me summarize, and then people jump in. One of the things they did was to were there earlier cases of COVID? So they did surveillance. They looked at records of morbidity, mortality due to respiratory diseases in and around Wuhan. National Sentinel surveillance. They have a pneumonia sentinel program in China, which picked this up originally. If there's more than so many cases of pneumonia, that would set this procedure in place. And that's because of the first SARS outbreak. They put that in place. They looked at laboratory confirmations. Interestingly, reports of retail pharmacy purchases for antipyretics, cold and cough medicines, a convenient subset of stored samples of research project from the second half of 2019 in various hospitals in Wuhan, the rest of the province, and other provinces. And they say none of them showed any impact. They looked at the disease reporting. So the samples were all negative for SARS-CoV-2. They looked at disease reporting, mortality, pneumonia-specific mortality from Wuhan and the rest of the province. They can see a big increase, the third week of 2020, but nothing before that. And, a number of people have said, well, this doesn't prove it didn't come from a lab. And I think, if there had been a lab outbreak, this would have picked it up. As Alan said, I think on TWiV a few weeks ago, if people started getting sick, this would have been an alert. So I'd don't think there's any evidence of an earlier signature. And they do say throughout the report, we need to get more records and check more and more and more because this is not all we have. But I don't see any signature of an earlier. And there are plenty of graphs where you see the rise in pneumonia, for example, in January, but nothing really before that. So they say, and then they have, all right, so the conclusion, unlike any substantial transmission of SARS-CoV-2 in Wuhan during the previous 2 months. And they went into the, go ahead.
I was just going to say, I think that what's important to note about that sentence you just said was no proof of substantial spread in Wuhan beforehand. Because I think that, if we think about different origin hypotheses and we think about the origin of lots of different emerging infectious diseases, sometimes we see emergence in areas away from the original point of detection.
Sure.
And so just because we first detected this in Wuhan doesn't mean that the first actual cases were in Wuhan.
That's right.
And so I think it's important that we point out that it shows that there was no earlier spread in Wuhan. It doesn't mean that, say, in some rural place or some other, wildlife farms, there wasn't something going on earlier.
Yeah, they clearly have to do an expanded surveillance. But if you think it began in the Wuhan Institute of Virology, I think there's no evidence from this.
Right, exactly.
Right, that happened. Unless you're going to say, well, maybe they were all asymptomatic. Okay, you can just keep piling on maybes, but it doesn't help the situation.
And I thought they had even done serological testing of the people in the laboratories. And so you would think that, maybe asymptomatic, but then also negative for antibodies.
Yeah, they have a lot of stored serum samples, and none of those were seropositive.
So I want to clarify something about the whole came from a lab idea, because there are two different stories. One is that it was created in the laboratory artificially somehow.
Right.
Okay. Engineered, and then either accidentally or deliberately released. And we and others have argued against that idea just based on the extreme unlikelihood that anybody would create anything that looks like this. We're not smart enough. Okay. And there's too many changes.
And also Amy made a point to that the other week that I thought was a phenomenal point, that what is the likelihood that you engineer a virus and then suddenly find its relatives in nature shortly thereafter?
Right. Mm-hmm. Good point. And so the other scenario is that since this is a laboratory that has been doing surveillance, broad surveillance for coronaviruses in other animals that involve sampling and bringing them back to the laboratory and sequencing them and maybe doing further studies on some of the more interesting candidates. The other story is that somehow this particular virus was isolated from somewhere in the wild or something close to it and recombined with something in a laboratory, but that's even extremely unlikely. Let's just go with the isolated in the wild and worked on in the laboratory and then somehow accidentally escaped either in an infected individual or otherwise. There's no record of anything like this, and none of the individuals involved in the work provide any evidence that this exists. So in order for that to be true, there has to be a conspiracy of people lying about this. All right. Yeah. Which I think is unlikely, extremely unlikely. And I would point again to what Alan said last week, is that the Mark Twain quote is that you can, three people can keep a secret if two of them are dead. All right. And there's been absolutely no indication at all that scenario number 2 is credible. All right. Yeah. Now, one problem with all of this is you can't prove a negative. And scientists, being what they are, will say, well, I can't be 100% sure, but I can put a likelihood on it. And I would say, personally, very unlikely. Somebody has to be lying in order for that to be true. And I'm not ready to go there.
Right. And the verbiage used in the report is extremely unlikely.
Yeah.
They talk a bit about the Huanan market. Initially thought to be associated, but they note there are a bunch of cases not associated with the market. There were a bunch of cases associated with other markets. So they can't really say if any of these markets were involved.
However, there were a lot. There was quite a number of surface samples from the Huanan market that were positive. So if nothing else, I think of that as being potentially a hot spot for, if nothing else, spreading virus.
Yeah, that's a market.
Clearly it was
Makes
there.
sense. Yeah.
Clearly it was there.
They had 923 environmental samples and 73 were positive.
Yeah.
So surface contamination. But they also had, what they say, 80,000. No, that's from China. But they had samples of some wildlife from the market, and they were all negative, right?
Right.
They also had 80,000 wildlife samples from throughout China and nothing positive for SARS-CoV-2 before or after the outbreak. And none of the animal products in the Huanan were positive as well.
My favorite sentence there is, there was no obvious clustering by the epidemiological parameters of exposure to raw meat or furry animals.
I love it. Furry animals, yes. That same paragraph, sequence data showed that some diversity of viruses already existed in the early phase in Wuhan, suggesting unsampled chains of transmission beyond the Huanan market cluster. So that's very interesting that it was clearly circulating somewhere. They also talk about the viruses that have been identified that are the closest. The RATG2 bat isolate from 2013 is the closest known to SARS-CoV-2. And there are others that are close. And they say neither of the viruses identified so far is sufficiently similar to serve as its direct progenitor. And so this is not, you could not make RATG2 in the lab and then, play with it and it gets out and causes SARS-CoV-2 outbreak. It's too far away to be the direct progenitor. So, a lot of people think that was it, and it's not. It's just not the progenitor. All right. The other thing that's quite interesting. So they're pretty sure that the outbreak started in December 2019, but there are reports from a couple of countries which they review that suggest that the virus circulated elsewhere. And I think one of them is in Italy, for example. And they say, so far, the quality of the studies is limited. And it's true. None of them have been confirmed. I think one is a serological survey, which is subject to, cross-reactivity, and the other is a PCR, which is subject to contamination. And they're just individual reports. I never thought they were very, very convincing as well. Let's see what else. And then they have a bunch of other studies they suggest. And they have 4 scenarios for introduction. And I will rank them in terms of likelihood of each possible pathway by the team's assessment. Direct zoonotic spillover is considered to be a possible to likely pathway, right? Direct zoonotic transmission from an animal to humans. Introduction through an intermediate host is considered to be a likely to very likely pathway. So that's next. Introduction through cold/food chain products is considered a possible pathway. And last, introduction through a laboratory incident was considered to be an extremely unlikely pathway. I should note, I forgot to do this, they do discuss some evidence that there were some outbreaks in China caused by contamination of frozen food packages or something like that.
Right. They call it the cold chain. And at first I was really confused by that. And then I thought, how did I miss all the discussion of this cold chain transmission?
Yeah. So it's, as far as I can tell, it's only in China, and I'm not sure. I don't think I've ever seen those data. Right. Mm-mm. So, but they do put it third out of the 4. And, I'm not sure that we'll ever get to the root of that. I mean, you can, if you do enough wildlife sampling, I mean, the idea is you're going to have to do a lot of sampling of wildlife in areas.
Absolutely.
And that includes China, but it also includes Thailand, as we discussed a few weeks ago. They got SARS-CoV-2-like viruses, also in Japan, and who knows where else. So,
right. So this, how long after the first case diagnosed in China did this team start working? How many months or a year, or what was the time frame? So
the first case was December 1st, 2019.
And this commission was founded when?
This commission was founded, I think, in May.
Exactly. So horses,
but they didn't
horses out of the barn. That's the only expression I can think of right now. If you go looking for them, they're going to be up in the mountains. They're not going to even be in the same city. So it's very difficult to find something of such an immediate event as a spillover. Somebody gets sick, and then after that, everybody is catching it. How do you do that 6 months after the fact?
Well, SARS-1
these are all
we found the likely progenitor of SARS-1 a couple of years after the outbreak, right? So
sure, but then what?
But you can't prove it, though. That's the point. So
no, that's true.
You can never prove that this was the progenitor because it's many years later.
Yeah, but that's the point. So this commission is after the fact. It is not even valid for the next outbreak.
I think you need to know the likely route of emergence so that you can say, oh, look, this is something we could change in some way.
And I think that this whole problem, in particular, the controversies that we've highlighted, it's not going to go away until some positive result comes up that says, here are data that say that, it's going to be a sequence of a virus from an animal that is unmistakably very closely related to this. That's what did it for SARS-1, right?
Yes.
Right. Yeah, there was an isolate from a civet, which was 99% identical, and that's likely, but you cannot say that was it.
What if it went the other way?
Because it was sampled after the event. It's not
exactly.
And I had this conversation in one of my classes last week about just how hard this is. And I think that sometimes people underestimate how hard this is. So I gave my students an example of, okay, imagine that you are an alien from Mars and you want to know if SARS-CoV-2 infects humans. So you come down in your spaceship and you pick up one human and you PCR them. It's entirely likely that you could have picked up someone who is either recovered or has not been infected. The probability of you picking up a person who is actively infected in your one person you pick up is quite low. And so if we're going to do this surveillance, it needs to be pretty extensive in order for us to conclusively say this virus is or is not in whatever population.
Yeah, and the surveillance is not easy. It's not easy because you need a lot of people, you need money, you need lab resources, and who's going to pay for it, right? If you go to the NIH in this country and try and do surveillance in bats, you never get funded, right? That's why it's hardly done.
So, Vincent, isn't the only major viral infection of humans that isn't underfunded is influenza?
I think HIV/AIDS is
HIV/AIDS.
Okay, but in influenza, at least, they can tell you the seasonality of a particular strain, right? And when the wild birds come to China and they land on the duck ponds and all this other stuff.
All kinds of wildlife surveillance of wild birds and pigs on farms and all that.
Yeah, exactly.
And it's global. It's a cooperative thing. It's not just the United States.
And because of the 1918 pandemic, I think that's why we have this.
Well, maybe.
Guess what? We got another one.
Yeah. Maybe something will come out of this.
Well, we're not looking in the right animals for this one, though. That's right.
Well, I think it's a bat or
I don't know. They're harder to catch than cranes.
I think the bat is the likely ultimate source. But if rodents could be, some of the coronas came from rodents. So you could look, but we have very little wildlife surveillance. You're right, for other than influenza. That's true. Yeah. And, when you think about the HIV origin studies, it took over 10 years of going to Africa where we said, okay, this is where it looks like the first cases arose because they have a 1959, serum that was positive, and they start looking in chimps.
Yeah, exactly.
And that, like 10,000 chimps later, man, that was a big deal.
10,000 chimps later.
Yeah.
And then finally, the conspiracy theories stop when they say, here it is, here's the origin.
Right. And again, remember that our first detected cases, our first published cases were in LA.
So also keep this in mind that we don't really know how many species of anything there are out there. So we're still discovering that. no less
and
their infections.
We did not call it the LA virus. Right?
We did not call it the LA virus.
Actually, it was called LAV, but that was different.
Different.
So, I think it's important to recognize that this could have come from somewhere else. But yesterday, a reporter asked me, why do people keep being fascinated with these theories? And I said, because getting the truth is hard, and it's not going to happen anytime soon. And so people want an explanation for this. But I also think it was fomented to a certain extent by the previous administration, right? By calling this the China virus and all that.
You think?
I mean, people want someone to blame.
They want to blame.
They do. And I remember their retort to this also. They said, no, it started in the United States. It became oh yeah, your mama. It got very bitter at one point.
There's also something very appealing in a negative kind of way about escape from a lab. It just it grabs headlines. All right,
it's true,
and people like that kind of stuff, but that doesn't lend it any credibility.
So one of the members of this committee was Peter Daszak, and I've emailed him multiple times to try and get him to come on TWiV, but he hasn't responded. Nevertheless, he was apparently on NPR this morning, Dixon, right?
Yes, he was, and I heard him, and he was particularly uninformative, I thought. I don't mean to sound critical, because Peter is a very nice man, and he wants to tell you what's happening, but...
I take issue with that.
You do? Okay.
Yeah, I thought it... there were two different spots on NPR where they talked about the WHO report, and I guess you heard the one with Peter, the same one I did. I did not listen a second time, but I think he again alluded to the wildlife farming that Vincent, you reported to us a couple weeks ago. I don't know in what venue he had presented or talked about that hypothesis, where there's these wildlife farms to increase the welfare of people in rural communities, and that. So he alluded to that being a very possible source. I don't know. It's a 6-minute listen. You can put the link in the show notes. People can.
Yeah, they didn't give him enough time to really say much.
He could have an hour on TWiV, and I told him when I wrote, I said, not only can we give you an hour, but we're always very nice to you. We don't challenge you. So when we're done recording, I'm going to email them again. And I'm going to keep doing it because it would be interesting. And Dixon, you need to be on that episode because you're buddies.
I would love to be. But he also very quickly pointed out the fact that he's been working with the Wuhan Institute of Virology for 20 years or something like that. Well, so he has a lot of contacts there. And I don't think he would ever support anything that said that that's where the virus came from.
Right.
Well, what if he had?
Because he knows it very well, and he knows that nothing could happen like that.
Yes, but if he had the evidence, he would say he wouldn't hide it. But he does. There's no evidence, right? I agree.
And that's exactly what he said: is that there's no evidence.
Yeah, that's that. Well, yeah,
and that's what the report says. There's no evidence.
Correct.
Yeah, but people who don't want to believe it will not believe it, right? That's right. They'll find fault.
There are a number of those around. There was the NewsHour had a report on this last night as well. They interviewed, among other people, David Relman, or is that his name? Yes, David Relman from Stanford.
Yes, that's right.
And I found it annoying.
Well, Relman has an agenda. He is a anti-gain of function person, right? And he's been involved ever since the H5N1 ferret experiments. And
I'm going to trust the guy. I'm going to trust the guy who was on the panel, who actually did the investigation, and who has done the surveillance in the wild, and who knows all of the characters involved.
So Relman's main comment was that there was not enough data in this report to make any conclusion. That's what he said.
I disagree. That's what he wants to believe, They will say, "Oh, it's an MD from Stanford." And this reporter I talked to yesterday, I've talked to her a lot, and she said, "I know this is an MD, and that doesn't matter, right?" I said, "No, it doesn't matter what you have after your name. If you don't know the field, you don't know what you're talking about." She said she got it. I talked to David Quammen yesterday as well, and he got a bunch of things right. And I said, David, it's very impressive. You seem to be one of the few, non-scientists that are getting this right. Because we were talking about fitness. And he said to me, oh, so fitness can be a lot of things, right? Not just one property. Yeah. Yes, David. So he's writing a book and he's talking to all kinds of people. Anyway, let's move to a few other things. Kathy, you had found this morbidity and mortality weekly report on the effectiveness of the 2 mRNA vaccines at preventing actual infection. Tell us about that.
Right. So it's just out and MMWR is a CDC publication. And the first word in the title is interim estimates. So I think that means that they're going to continue to gather data. It's from 8 U.S. locations from frontline workers from December 2020 to March 2021. So very recent data. And 4,000 or so participants, and they looked at the main takeaway for me is that they're going to demonstrate effectiveness of these vaccines. And what you remember is that from the clinical trials, we get efficacy. And now that we're getting real world data from these healthcare workers or frontline workers, not just healthcare, I think they included, for instance, grocery store workers and primary responders, front and so forth. Anyway, that from this real world population, it looks like there's about 90%. They could not disaggregate the data between the 2 mRNA vaccines. But what they did also, I didn't highlight this. They they had people do self-sampling quite frequently. I think it was at least weekly.
Yeah.
And then send those in. And so they were able to then do qPCR on those samples to look for virus. And help me out here.
There's 1.38 infections per 1,000 person days in unvaccinated and 0.04 infections per 1,000 person days in the immunized, partially and fully immunized.
1.38 versus 0.04. So the immunization seemed to be quite effective, and the estimate is on the order of 90%.
For full and 80% for just one dose, right? Yeah.
Right.
And this is against infection and not just symptom.
That to me is the most important thing, because up until now, most of the data is relevant to symptomatic disease. And one of the reasons that we've been recommending that vaccinated people continue to wear masks is that we didn't know whether vaccinated individuals, even though they don't develop symptomatic disease, might actually get infected, replicate virus, and maybe even transmit it. And now this is real life data that says that there is a very significant 90% reduction in whether you actually even get infected. Now, that to me doesn't mean I'm not ready to recommend that all vaccinated people take their masks off, okay? Because I want to follow protocol here, but it's very encouraging news, and it's a distinction that the news media don't make. Okay, there have been, I've seen news reports on this that say there's new data out there that show in the real world there's 90% effectiveness of these vaccines, but they fail to point out that this is effectiveness measured by a different and more stringent criterion, really. It's whether or not you actually get infected, not just whether or not you get symptoms. And so, to me, this is great news.
All right, I totally agree with you, Rich.
I think the other take-home is also that it shows the 2-dose immunization is really important, right?
Mm-hmm.
You get better, you get 90% as opposed to 80, so you get both doses.
Is there any data on Johnson & Johnson?
No,
so that's pretty popular
not from this.
Right now.
Not similar, no.
That's going to take a while.
Yeah, okay.
Yeah.
I'm sure there will be.
Oh, yes.
There were 5 participants that had received the Johnson & Johnson vaccine, but they don't disaggregate that set of data either.
No.
Yeah.
Is anybody going to mention AstraZeneca's double report?
Yeah, we're going to discuss it now. Yeah.
Okay. Good.
So Rich, you sent this link to I don't know, it's a manuscript, I guess.
It's a preprint.
It's a preprint.
Preprint.
That's on a preprint site that I'm otherwise not familiar with. Let me see here.
Research Square, right?
Yeah. Mm-hmm. Titled a prothrombotic thrombocytopenic disorder resembling heparin-induced thrombocytopenia following coronavirus 19 vaccination. These are the data about which we have heard reports recently. Okay, and I think I also link here to a science magazine write-up that I think it's the same write-up that we talked about last week, where they allude to this, but these are the actual data. Mm-hmm. So this is a group in Germany, the Greifswald Medical University, and actually McMaster, that's in Canada, Paul Ehrlich Institute, that's in Germany, I think in Munich, and Austria as well, Medical University of Vienna. And they report on this clotting disorder in 7 individuals, or was it more? The table shows
8.
9. 8?
Oh, well,
9.
patients,
the table shows 9. 9 individuals within 2 weeks after vaccination, and they give a bunch of data about the patient profile, and they report on the characteristics of the disease, and most importantly, so these are clots in different places, hepatic vein, some cerebral clots, etc. 4 of these individuals died. The others are recovering. And what they find about this that to them is unusual is that 4 of these, at least, show antibodies to, these are autoantibodies, to a platelet cytokine that's involved in signaling for clotting. And presumably the idea is that these antibodies, in reacting with this cytokine, dysregulate the clotting cascade, and that causes these people to throw off clots. And it seems to me that the main argument here, and we've talked before about the fact that the European Medicines Agency, in looking at all these data, said, look, the amount of clotting disorders in the vaccinated population does not exceed the number of clotting disorders in the general population. So as a general consideration, the risk here of clotting after vaccination is there's no signal, okay? What these individuals are saying, I think, is that this is a very specific and rare and unusual clotting disorder that's occurring at a higher frequency, it appears, in the vaccinated population than the unvaccinated population. And I have to say that in reading through this, they quite glibly call this vaccine associated or actually vaccine induced.
Yeah.
Okay. And so they, to me, aside from the fact that there's an association in time with the vaccination, there's no convincing proof in any of this that the vaccine is actually doing this. However, it's something that I think bears watching.
Yeah.
And we will find out. I think we need more data over a longer time from more places. Okay. And so keep your eyes peeled. I don't think this is an indication I don't think it's the bottom line necessarily, but that's the story as I understand it.
So people who have long-term infections and go to the hospital and develop complications after they go home, do any of these people get this exact same syndrome?
If they do, it's not mentioned here. And I think there's a lot of questions that I have in this that are for Daniel. Okay.
You're right. Exactly.
There's a lot of clinical stuff in here about clotting that I don't frankly understand. Okay.
Yeah, I mean, the patients do get clotting disorders with COVID, Dixon, but I'm not sure they're the same
not
thing
this
here.
kind of clotting
Not
disorder.
this kind of clotting disorder, correct.
So
or maybe they haven't been sussed out in the huge population of clotting disorders.
Could be.
I mean, they had to take their serum here and do these reactions, right? And maybe they haven't done that,
Right.
So
but what they do say here, which is, I think, interesting, right? They're saying that these disorders resemble heparin-induced thrombocytopenia, but with a different serological profile, which to me says they don't usually see this, right? Mm-hmm. So that could be the only unique aspect of it, and maybe. But as you said, Rich, last week, these can be treated, right?
Yes.
So now that we're on the lookout for it, and I do want to mention that, so this is 9 cases, and there have been 55 million vaccine doses administered in the EU, so...
Right.
If it is cause, it's pretty rare if it is. And
as we talked about last week, so far at least, as far as we know, this is only seen with the AstraZeneca vaccine. Mm-hmm. Now it could be that now that people are awakened to this, that it'll show up elsewhere. But it does raise the question: if it is specific to AstraZeneca, what is it about that vaccine? If it is vaccine induced, that's doing this, and that Kathy, remains to be seen.
They mentioned this fact that some ads bind platelets, right? So what do you think? How could that be involved, or could it?
I think it could be if I don't know. It's a guilt by association kind of thing. I don't know.
So do many ads bind platelets, or just a few? Or
I couldn't tell you. I'm not up
maybe
on that.
So maybe chimp ad, as you said last week, someone said it does something differently. Who knows? Or maybe it's the prefusion spike also. Not prefusion spike. Who knows? But
to refresh my memory, please. All of the proteins that are being induced in our cells by these vaccines, is that the same from, let's say, Moderna and Pfizer? I think it is, but is it also true for AstraZeneca's protein that it's inducing? Is it exactly the same?
No. But my understanding is, correct me if I'm wrong, my understanding is that as E1A knockouts, so the mRNA vaccines are just spike.
Right.
The J&J vaccine is a recombinant with Ad26. The AstraZeneca is a recombinant, spike-expressing recombinant with a chimp adenovirus. In the case of the chimp adenovirus, it's not a prefusion stabilized spike, but other than that, it is the same spike. Okay. My understanding with all of the adenovirus vectored things is that since they are E1A knockouts, that there's going to be very little, if any, gene expression from the virus during the infection, and that it's going to be primarily the spike protein.
What is driving
CMV
spike
promoter?
CMV, okay.
Okay, it's an exogenous promoter. Okay, so to me, if there's an antigen that's unique to a given vaccine that is causing some sort of side effect, it would be a structural protein involved in the virus that's delivering
right, that's why I asked.
The DNA. Now, Kathy, can you comment on the E1A thing? Do I have that? Can you confirm that it's going to be very little, if any, actual viral protein gene expression in those infections?
Yes, I think I can confirm that. I do know that if you have very high multiplicity adenovirus infection of an E1A mutant, you can get some gene expression and viral replication, but it's very, very high multiplicity of infection. So that, I don't think that's great.
My understanding has been that there's going to be very little gene expression, if any, close to undetectable.
Folks, rewind the last 5 minutes because there's good information that mostly is is what virologists know. Okay, that's what we do.
Right.
And you can understand why there are no viral proteins produced.
Right. I did look up adenovirus platelet interaction in blood causes virus sequestration to the reticuloendothelial system of liver.
That's good. It would clear it then.
2007. Yeah. Virus platelet aggregates are taken up by Kupffer cells and degraded.
Kupffer. Love that word.
Back on these guys calling it vaccine induced. So they're the ones who coined this term VIPIT, vaccine induced prothrombotic immune thrombocytopenia. So at the very least, it should be VAPIT, just vaccine associated.
Yep, I agree.
Yeah.
I think that the biggest potential danger of this whole thing is people losing confidence in the AstraZeneca vaccine.
Here.
That's the biggest problem.
I would agree.
Now, we can keep an eye on this. We can deal with it. Okay. Maybe it's true. Maybe it's not. The final word isn't in, but the vaccine is already, we know, is of enormous value and globally.
Right. And as we said a few minutes ago and last week, there are treatments for it. This paper does describe those as well. Therapy with non-heparin anticoagulants.
Right.
If these signs or symptoms appear 5 to 14 days after vaccination.
All right, so let us do some picks.
Oh, good.
It was a heavy episode.
I love it.
A lot going on. There's a lot going on.
This is where ecology crosses the nexus between all the other sciences, and I just loved it. It was fantastic.
Dixon, you know so much about sewage in New York.
I do.
Where does all that come from?
Dixon,
Well, I'm busy designing a new city. No, my classes are now designing a new city, and part of it is water harvesting. So you have to know, why are you harvesting water? And the answer is because we often get water shortages, but we're throwing away 1.2 billion gallons of water per day. Why don't we just recycle it? I don't know. I don't understand.
Yeah, well, you don't have to drink it. You could just use it in the chillers, right?
Of course. Or you could ship it back to the reservoir and nature will recycle it for you. There's a lot of options here, but we discard it, and we spend billions discarding it. At any rate.
What do you have for us, Dixon?
I love this video. My little grandson is only 5 years old, won't stop watching it. You won't either. It is 250,000 dominoes put together by numerous teams inside of a gymnasium. And then you push the button for start, and all you hear are the tumblings of dominoes. It's absolute dead silent, because they want to hear their own structure that they've built fall into little pieces. And it's sequential. And this thing, wait till you see the end. Watch it to the end. Don't get discouraged, although you won't, because you'll be fascinated by all the little things that you'll see some common cultural references to Mario, Marvelous Mario, and all those other things. But at the end, they're timing this thing. Imagine 250,000 dominoes lined up, and they can tell you exactly when it's going to end. When they start the first one.
This is Rube Goldberg on steroids.
Yeah. That's another way to put that.
Amazing.
There's a little Rube in there, but it is, if you want something to do when you're up at 3 o'clock in the morning and you don't want to go back to sleep, this is a wonderful thing to do.
I got to send this to my grandson.
It's so much fun.
I already sent him my pick for the week.
Nice. Dixon, you just found this for your grandson. You're not hanging out on YouTube, are you?
No, who, me?
I didn't think so.
Depends on the YouTube.
Brianne, what do you have for us?
I have a video also on YouTube, but originally from Cold Spring Harbor DNA Lab. That is all about the science behind the RT-PCR COVID-19 testing. And so many of our listeners may kind of have caught on to how this works so far, but this goes through all of the details of how RT-PCR works, and it is just fantastic. It really covers every detail in a way that I think a lot of lay people can understand. I was incredibly impressed when someone sent me this video. So I think it's a really nice
Excellent.
Video.
That's good. Good to have that. Yep. Rich, what do you have?
I have an article from IFL Science that shows up on Facebook called "Beaded Lacewings Can Take Down 6 Termites with a Single Fart." And this relates back to a study that dates back to 1981. And it links the Wired article from, I forget when it was, in the 90s sometime, that is, a similar description of the same study. The beaded lacewing is, it's a flying insect. That's all I can say. And apparently the deal is they lay their eggs near a termite's nest, and the eggs hatch into larvae, which then creep into the nest. And these larvae can rear up and expel some sort of vapor from their rear ends that has the effect of paralyzing any termites in the vicinity. Okay,
probably cyanide based.
Up to 6 of them. They don't know exactly what it is.
Yeah,
and then the larvae proceed to eat said termites. Okay, and the termites, of course, are eaten alive. Nature being so inelegant. All right, so this gets headlined as "Take down 6 termites with a single fart," which I figured my grandson would love. And
no question about it. No question about it.
And when you just barely click on the link in the Google Doc, it cites the New York Times bestseller, "Does It Fart? The Definitive Field Guide to Animal Flatulence." So there's even more.
Yeah.
It's one of Rich's nested
bathroom
picks.
humor at its best.
Apparently, that's a New York Times bestseller.
Yeah.
You got it. Again, the guy lives in Flushing. What can I tell you?
People are obsessed with farts, aren't they?
Yeah.
Well, it's amazing how frequent they occur.
Kathy, what do you have for us?
I have show and tell so anybody who's on the video can see this 2-page story from the Sunday New York Times Magazine on which you have the entire SARS-CoV-2 sequence of the B117 variant, in fact. And there's white spots where there's mutations. And so this is from an article by John Gertner, a freelance writer. Starts out talking about Eddie Holmes and Yanzhen Zhang, talking about releasing the SARS-CoV-2 sequence in early January 2020. The bulk of the article is about genetic sequencing and that and how it's not just technology, but it's platform. It's a platform innovation that revolutionizes marketing and society, sort of like cell phones and web browsers are platform innovations, not just technology.
Cool.
And it has a fact in it that there's a min-ion sequencing on the International Space Station. So we were talking about min-ion sequencing earlier in the show, and that came up. And then also talking about, we've probably heard about the possibility of the $1,000 genome, and actually they think it's going to be $100 to do a complete genome sequence, and maybe with a new chip that's being developed as low as $10 before very long. So it's a really good article, I think easy for people to read, and you could read it online, but you might want to get this hard copy so you could save the sequence, and you maybe have a friend or neighbor that's still working on the Sunday Times crossword or the Beehive puzzle, and you can still get the hard copy.
The Beehive puzzle's good.
Right.
Yeah, Dixon, you got me hooked on that.
Yeah.
Oh, the TWiVers. Do you know how many words come out of that?
Yeah.
It's incredible. It was one of the biggest spelling bees ever. By the way, Kathy, I'm reminded of Gattaca. Are you not when you see something this happening here?
Yeah.
Interesting movie.
All right, I have a pick, which is a page from a website called immunize.org, which looks like a reasonable website. It is about administering vaccines to adults, dose, route, site, and needle size. There's no SARS-CoV-2 on this, but someone asked me last week, isn't 500,000 an awful lot for SARS-CoV-2 vaccine? I said, no. So I sent them this, where you can see the dose ranging from 0.2 to 0.5 to 1 ml. Which seems to be the upper limit. What kind of route of inoculation, how to do IM, how to do sub-Q, even how to do flu mist in the nose, and needle lengths depending on your size. I didn't know they needed to gauge, so they must look at you before they immunize and pick the needle based. Dixon, do they ask you what what your weight was before they injected you?
Not even, no, they wouldn't dare. But do they also list how to do an intrathecal injection?
No, they don't. They don't list how to do that. So if you are less than 130 pounds, you go 5/8 to 1 inch. And if you're over 260 pounds or 200 pounds, a 1.5 inch needle, which is quite long.
No,
that's for IM, of course. For IM, you have to get deep into the muscle. Anyway, it's kind of a nice collection of factoids. It's a PDF, so you can save it.
You might say that's a deep dive into vaccination.
Yeah, it is a deep dive. Arg. But there, if you want to know how many mLs or what kind of needle, there you go. All the different vaccines. They should update it with SARS-CoV-2 vaccines, right?
Indeed.
It'll be taking up the whole chart. There's as many of them as are. All right, that's TWiV 737, the number of a very famous airplane, a jet, right? That had some issues in the last few years, at least one version of it. The website for the show notes, microbe.tv/TWiV. The email for sending us your questions, TWiV@microbe.tv. If you want to send them to Daniel, it's Daniel@microbe.tv. Don't send them to TWiV, please. If you'd like to support us financially, please go to microbe.tv/contribute. Dixon Despalmier is at trichinella.org, thelivingriver.com. Thank you, Dixon.
Thank you, Vincent. A wonderful tour de force through the eco-virology of New York City.
Man, we need to get a title for this. That's going to be interesting. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Brian Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.